Hello, my name is Chelsea Jackson, and I'm representing group number 31 for the chemical, mechanical digestion, and absorption of the macronutrients. Let's get started. Starting with carbohydrates, mechanical digestion. Once food enters the mouth, mechanical digestion begins. Chewing physically breaks down food into smaller pieces so that it is easier to swallow and digest. Food then travels down the esophagus and into the stomach. Peristalsis, which are muscular contractions occurring every 4 to 5 seconds in the stomach, help to push through the GI tract. Once chyme exits the stomach, it enters the small intestines, furthering the mechanical digestion. The small intestine segments sections of the intestinal wall through muscle contractions in a process called segmentation. These contractions continue to break apart food, divide and mix chyme. Chyme then enters the large intestine, and through slow peristalsis, waste products are moved to the colon. Chemical digestion. Salivary amylase in the mouth breaks down starches into polysaccharide, maltose, which moves into the esophagus. There is no chemical digestion in the esophagus. The esophagus carries maltose into the stomach. The stomach inactivates the amylase, and proteases destroy the enzyme. Therefore, there is no chemical digestion of carbohydrates. Once the carbohydrates reach the small intestine, pancreatic amylase breaks down starch into more units of maltose. Further down the intestinal tract, microvilli split disaccharides into monosaccharides. Once carbohydrates reach the large intestine, bacteria ferments fiber or resists starch into gas or short-chain fatty acids. Once carbohydrates are completely broken down into monosaccharides, the small intestine takes over from here into the absorption process. The microvilli lined on the inside of the small intestine are responsible for absorbing the monosaccharides into the cell membrane. Facilitated diffusion carries the monosaccharides to the liver and muscle. Galactose and fructose are converted to glucose while they travel through the liver by the portal vein. Glucose and galactose and fructose are the final products of absorption. The liver stores and uses glucose to maintain glucose levels as a molecule called glycogen. Lipids. As lipids are not water-soluble, their digestion occurs somewhat differently. Mechanical digestion begins in the mouth with the act of chewing and moistening with saliva. Via the esophagus, partially digested lipids are transferred to the stomach, where churning movements such as peristalsis and segmentation further the mechanical breakdown of lipids. Peristalsis and segmentation continue in the small and large intestine, moving the bolus and undigested matter to the cecal colon to be expelled from the body. Chemical digestion of lipids also begins in the mouth, with the lingual lipase released from the salivary gland beginning the emulsification process. Once in the stomach, an enzyme called gastric lipase joins during the churning actions of peristalsis, which allows the lipids to be dispersed. This process breaks triglycerides into diglycerides and free fatty acids. About 30% of triglycerides are broken down in the stomach. As the lipids pass into the small intestines, their presence stimulates the gallbladder to release bile and the pancreas to release pancreatic lipase via the bile duct. Bile salts work with pancreatic lipase to further break down the remaining triglycerides. Bile emulsifies by breaking the globules into small pieces while keeping them dispersed in a solution, while the pancreatic lipase takes off one fatty acid at a time. The final products after these processes are free fatty acids and monoglycerides and glycerol. Absorption. Absorption of lipids occurs in the small intestines after the mechanical and chemical processes have finished. In order to make the monoglycerides water-soluble, bile salts surround the globules, creating what is called a micelle. Micelles transfer monoglycerides and long-chain fatty acids to the surfaces of the intestinal villi, which are immediately absorbed by the intestinal cells. Bile breaks off at this point and returns to the small intestine. After being absorbed, the monoglyceride and free fatty acids rejoin to become a lipoprotein. These complex molecules then exit the intestinal cells, becoming a chylomicron, which moves into the interior of the villi to be absorbed into the lymph system and eventually make their way to the bloodstream. Because short-chain fatty acids and glycerol are more water-soluble, they move directly into the bloodstream and after absorption from the intestinal cells. When the pancreatic lipase acts on the lipid, it breaks it down. This results in two digested products of lipids, free fatty acids and monoglycerides. These products are much easier for your small intestine to handle, and they have very little trouble being absorbed out of your digestive tract. It is worth noting that fats are reassembled in the epithelial cells of small intestine. This makes fat absorption different than absorption of proteins and carbohydrates, because with protein and carbohydrate absorption, their amino acids and monosaccharides are able to pass through the intestinal epithelial cells without being altered. Lipids also help transport fat-soluble nutrients such as vitamins A, D, E, and K. They contribute to the flavor and texture of food, and they are also an efficient energy source. And they provide the body with insulation for heat, as well as protect vital organs between ribs and hips that store fat on the tummy and protects as an organ interact with receptors to produce satiety. Now to proteins. The first step in the digestion of proteins involves the mechanical breakdown of these proteins through mastication. This occurs in the mouth. This mechanical breakdown allows proteolytic enzymes to work on single proteins. Next, the food is passed through the esophagus to the stomach. The stomach churning occurs, breaking down food into smaller pieces so enzymes can further break down the food. The small intestine is responsible for segmentation, the moving back and forth of food in order to further break down proteins. Peristalsis, which is a contraction of muscles, moves chymes through small and large intestine. The large intestine moves waste to the colon for expulsion. Chemical digestion. Once food is moved to the stomach, hydrochloric acid in the stomach kills pathogens and further breaks down plant and animal proteins. This acid also affects the tertiary and secondary structures which allow enzymes to break down peptide bonds. Pepsin, in a pH balance of 1.5 to 2, breaks down peptide bonds in the stomach. Once the chyme from the stomach reaches the small intestine, enteropeptidase causes the conversion of tryptogen to trypsin. This causes buffers to increase the pH to 7 to 8. Now pancreatic proteases can do their jobs and break down peptide bonds. Trypsin, chiromyotrypsin, elastase, and carboxypeptidase perform these jobs, and each of is responsible for breaking down a specific peptide bond. Epithelial surfaces in the small intestine can also contain enzymes like dipeptidases, which also break down peptides. What is left after this work is done are free amino acids, and through facilitated diffusion and co-transport, they are absorbed into the body. Protein absorption. Protein, once digested, finally gets absorbed as it makes its way through the end of the small intestine. This area of the small intestine is made up of the jejunum and the ileum. Lining the walls of the small intestine are very small finger-like mechanisms called villi, which have a small finger-like mechanisms on them called microvilli. The villi and microvilli act as grasping hands in the sense that they latch onto amino acids as they float by. Once the villi have latched onto the amino acids, they are ready to pass through the epithelial cells of the small intestine and into the bloodstream. One of the important things that was touched on earlier is that amino acids remain completely intact as they pass through the epithelial cells. Amino acids are the building blocks for the body, so of course they need to remain intact because after all, shape equals function. The final products of protein. The final products that come from the absorption of protein are singular amino acids that are processed and ready to link up to be used whenever might be necessary. The human body uses 22 amino acids altogether, and each serves a different function. However, amino acids are not only used as building blocks. In very extreme circumstances, where the body has exhausted all of its energy reserves, including carbohydrates and lipids, amino acids can be converted into energy, yielding glucose. This is truly a last resort and has very negative effects on the body, but in the worst case scenario hits, amino acids can be converted into energy. Here is the summary of how we broke down the presentation. Hope you liked it and have a great semester.